Hello everyone, and welcome to this audio podcast. This audio podcast is part of a Phantom edition. This audio podcast is part of a Vertex Roulet. This audio podcast as part of a Robotics Machina, and this audio podcast as part of a Storm Robotics production. In this audio podcast, a look at a research poster works that have been put forward for the International Pediatric Transplant Association IPTA in 2022. And This research poster presentation, this collaborative research poster presentation, looks at transplant immunobiology, and looks at transplant immunobiology and a chronic kidney disease and a chronic kidney disease review. The research poster presentation was collaborative between between uh, between uh, three academic institutions. Four academic institutions in the United Kingdom. And so to begin, some background perspective. So the um, the immune system protects the body from infection by pathogenic organisms. It is comprised of a complex constellation of cells, organs, and tissues arranged in an elaborate and dynamic and dynamic arranged in an elaborate and dynamic communications network. The immune system is, in its simplest form, a cascade of detection and adaptation, culminating in a system that is remarkably effective. Leukocyte and lymphocyte migration or trafficking from peripheral blood into affected tissues is an essential component of the inflammatory reaction to virtually all forms of injury, and is an important factor in the development of many diseases. Advances in the past few years have highlighted the central role of a family of chemotactic cytokines called chemokines in this process. Cellular adhesion molecules, or whether abbreviation or acronym is that's CAMS, have also been important in and where in and around in and where in and around where allograft rejection is where in where allograft rejection is evidenced, and as these molecules also allow protein receptor binding. Chemokines help to control the sort of selective migration and activation of inflammatory cells into the injured tissue graft. Chemokines and corresponding receptor ligands migrate during inflammation, and there is some evidence that certain negative states, such as, for example, duress or stress, duress or stress, and or for example anxiety, can directly affect physical immunity through production of stress hormones such as catecholamines and glucocorticoids. There is some indication that negative states may lead to a faster progression in certain diseases such as human immunodeficiency virus or HIV, and specific cardiovascular implications through direct biological mechanistics. Negative states may also indirectly affect disease processes through influence on health on healthcare on healthcare on on healthcare behaviors. So, for example, anxiety in some contexts has been related or regarded to many risk factors for poor health. Stress with respect to or regard to Hansen's general adaptation syndrome from 1936 has also been considered with commencing various disease states. So healthcare also incorporates another model called the biopsychosocial model. Biopsychosocial model, and the biopsychosocial model is also a sort of a technical term for the popular concept of the mind-body connection. The model was developed by psychiatrist George Engel at the University of Rochester as and the first published in a 1977 article of Science. The biopsychosocial model draws a distinction between the actual pathological processes that cause disease and healthcare service users' perception of health and the effects on it called the illness. These are also There are also theories that directly affect the immune system, and there are investigations that show this to be the case. And where collaboratively investigations are themed under 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 a specialism that actually where a specialism links to three different uh, different healthcare divisions. For example, where the main theme main specialism is referred to as psychoneuroimmunology, and the three divisions the three healthcare divisions include psychology, neuroscience, and immunology. So now looking at sort of the aims and objectives in regard in as part of this works. So, looking at the entities based on chronic kidney disease or CKD, and looking at transplant immunobiology and neuroendocrinology, these these entities form the basis of the literature and the works surrounding this research poster presentation in context of transplant immunobiology and a chronic kidney disease or CKD review. So, looking at the literature, looking at the literature in regard to transplant immunobiology, and looking at transplant immunobiology as a retrospective. So, there have been several investigations exploring or investigating clinical and experimental observations that indicate that immune responses may be modulated by the central nervous system or CNS. There was works by Niveau and Limaul in around the 1990s who highlight that the immune system and central nervous system are known to communicate through the endocrine and autonomic nervous systems, and inform that complete regulatory loop between immune and neuroendocrine systems exists. There was works by Wigant and team in 1990 who informed that there is a molecular basis for bidirectional communication between the immune and neuroendocrine systems. The recognition of such stimuli is converted into information in the form of lymphokines or lymphokines, monokines, and hormones, and a message received by neuroendocrine tissues. Nervous system recognition of stimuli can also be converted into chemical signals that can be relayed to immune cells, resulting in physiological changes. Works by Bedofsky and Del Rey in 1991 inform interactions imply that existence of different messages derived from the immune system, such as lymphokines and monokines, are capable of integrating immune neuroendocrine circuits. At the time of the peak immune response to innocuous antigens, as well as following inoculation of infective agents, endotoxins, or tumor cells, immunological cells release cytokines or cytokines which can stimulate the pituitary adrenal axis. The acute effect of these cytokines is mediated by the stimulation of the release of corticotropin releasing factor from the hypothalamus. Works by Grokowitz and team in 1991 highlight that conditioned immunosuppression using a taste aversion paradigm had been demonstrated in several laboratory investigations. The team report that survival of cardiac allografts in rat models can be in rat laboratory models can be prolonged by by immunosuppression using, for example, cyclosporin A or CSA as a stimulus in taste aversion conditioning. Conditioned animals received in a part of the investigation, conditioned animals received saccharin as the conditioned stimulus, paired with an injection of CSA at 10 and 6 days before transplantation. Early investigations suggest that behaviorally conditioned immunosuppression may have important clinical implications as an adjunct to drug treatments in transplant immunobiology. Grotzel and Sridharan in 1992 informed that integration between several immunological functions in host defense may reveal a novel mechanistics or mechanisms of cellular communication and biological adaptation. Ottaway and Husband in 1992 inform that many of the alterations in immunity resulting from central nervous system CNS activity can be explained in terms of changes in lymphocyte migration or changes in lymphocyte migration in response to endocrine signals, neural signals through neurotransmitter release, and or direct contacts between nervous and immune system. Kropunigs works in 1993 research suggests that the immune system is active even in a bidirectional feedback loop. In works by Klein in 1993, Klein highlights in 1993 a cellular molecular basis for modulation of host resistance is not fully understood, but involves altered functioning of both T lymphocytes and cells of the hypothalamic pituitary adrenal axis. Klein in 1993 also highlight that there is a production of cytokines and hormones produced by immune system and central nervous system, and works by Wagner and Blalock in 1995 inform or highlight that prospective investigations should seek to investigate into the physiological role that cytokines and cytokines and neuroendocrine hormones have between immunology and neuroendocrinology. Works by Perner and team in 1997 informed that in terms of cardiovascular, endocrine, and immune responses, acute high intensity physical activity, for example, and stress may be considered as a subgatory of stressful active coping. Neuroendocrine responses include increases in catecholamines, as well as elevations in cortisol under high but relatively low sympathetic activation. Immune responses include increases in natural killer cell or NK cell number and cytotoxicity, and suppressor and cytotoxic lymphocytes. Chronic life stressors influence active cardiovascular, endocrine, and immune responses to acute stressors. Physical activity also causes an increase in beta endorphin, and because of the beta endorphin, important immune units stay blunted. But in pre-transplantation, during stressful periods, or during stressful durations, there is no beta endorphin. So immune components such as chemokine receptors, CD8 T cells, natural killer cells or NK cells may influence acute allograft rejection post-transplant. As works by Frederick and Stranova in 1997 inform that neuroendocrine system influences and or modulate the immune components that link to physical stress. Works by Danza in 1997 informed that there is evidence that different perceptions of stress have different physiological consequences, different ways of coping, where there's different ways of coping with stress result in different consequences on immunity by nature, and outcome of which depend on the type of immune response. Webster in 1997 demonstrated that glucocorticoids and catecholamines favor T helper type 2 cells over T helper type 1 immune cells and mediators by controlling the production of a specific regulatory cytokines. In works by Gillard in 1998 informed that immune neuroendocrine that informed that immune and neuroendocrine interactions are involved in numerous physiological and pathophysiological conditions, and seem to play an important role to maintain homeostasis. The mutual information circuit inside of the immune, nervous, and neuroendocrine systems suggests that there is a link with pivotal regulatory role in living organisms, pathophysiological conditions, and seem to play an important role to maintain homeostasis. The mutual information circuit inside of the inside of the immune and nervous and endocrine system suggests that there is again a link with pivotal regulatory role, where there's pivotal regulatory role in living organisms, and if in works by Ferencik in 1998 assert that the immune system is much like a sixth sense, and may ultimately provide a new understanding an understanding of physiology required for disease and stress involving immune neurocrine and immune neuroendocrine communication. works by Clapp Heath in around 2000 inform that though there are questions that remain unanswered, where there are questions that remain unanswered, it appears reasonable to include that anxiety, and possibly stress factors may affect the use of immunosuppression in transplanted recipients. There's works by Hennig and team in 2000 who carry out an investigation on the mechanism of changes in lymphocyte numbers after stress. The team identified catecholamine is linked to stress and found it is of major importance surrounding frequency of myeloleukocytosis. In another investigation by Hennig and team in 2001, demonstrated that cortisol seems to play an important role in stress-induced redistribution patterns of CD8 cytotoxic T cells. This, however, can be explained by different migration processes between cells, for example, and different targets of migration, and therefore different glucocorticoids influences on target tissues. In works by Gillard in 2001 informed that crosstalk between immune and endocrine systems is important to homeostasis, since the interactions can produce various appropriate adaptive responses when homeostasis is threatened. In works by Harbers in 2003, the high the where Harbers in 2003 highlight that in the field of psychoneuroimmunology, hormones in the field of psychoneuroimmunology, hormones, peptides, and neurotransmitters regulate the immune system. However, understanding the complex interactions between systems is still a challenge for the 21st century. Pathogens, tumors, and allergens are detected with great sensitivity and specificity by the immune system. However, George Engel's biopsychosocial paradigm is now one of the main models, dealing with most human disease, most human human conditions. The biopsychosocial model also encourages reading into areas not traditionally associated to transplant immunobiology. Blanlock's works in 2007 also informed that as a sixth sense, the immune system is a means to signal and mobilize the body to respond to these types of challenges. Investigators should acknowledge that research surrounding transplantation should not be restricted to single organs, and that the psychoneuroimmunology should not be restricted to single organs, and that psychoneuroimmunology may have a role in prospective investigations. This may produce a better understanding of the immune system, and it may be a means of attaining T cell tolerance post transplantation. Now looking at the principles of chemokine action, and of principles of chemokine actions, and where the principles of chemokine actions is sort of is in as part of this research poster presentation is adapted from works by Segreger adapted by works by Segreger and team from 2000. So the principles of chemokine action include where chemokines are released by stimulated cells, are released by stimulated cells, sort of as locally in context of pathophysiology. Chemokines are produced by intrinsic cells as well as infiltrating cells. Chemokines act through G protein coupled E7 transmembrane receptors and induce calcium influx. Chemokine receptor expression is cell specific. Cell specific. Chemokines bind to proteoglycans and extracellular matrix. Chemokines induce haptotaxis, where there's cell migration along surface gradients, and chemokines can activate adhesion molecules. Chemokines can induce oxygen burst, and the release of granules and proteinases by leukocytes. And chemokines control aspects of immunomodulation, angiogenesis, hematopoiesis, and development. Now, as part of this research poster presentation, I look at sort of prospective investigations. So works by Al-Arabi and team in 1997 assert that pre-transplantation plasmapheresis, together with immunosuppressive drugs, so for example cyclophosphamide and prednisolone, can be helpful in amongst renal transplantation. It can be helpful in the removal of human leukocyte antigen HLA in amongst immunized recipients awaiting renal transplantation. It can be considered a valuable approach to increase the probability of successful transplantation. In one investigation by Nafar and team in around 2006, there is suggestion where plasmapheresis with or without other therapeutic measures may have a role in the salvage of allografts with early acute dysfunction where recipients may be resistant to conventional therapy. The team conclude that allograft prognoses in recipients with acute humoral response who respond to plasmapheresis can be comparable to that in other renal transplanted recipients. In vitro investigations using interleukin 15, interleukin 2, and interleukin 15 receptor alpha, so IL-15, IL-2, and IL-15 receptor alpha, stress cellular adhesion molecules, and relevant monoclonal antibodies, so for example anti-CD25, could be investigated with chemokine receptors such as CCR5, CCR7, and CXCR3 in a T cell culture system to see if this affects T cell migration post-transplantation. Using peripheral blood mononuclear cells, or PBMCs, would also be advantageous. So in sort of discussion and conclusion, as part of this research poster presentation, this research poster presentation from 2022. So whilst there have been some collaborative approaches when between disciplines and practices which incorporate laboratory investigations, understanding CKD in context of transplant immunobiology is pertinent. Indeed, stress adaptation and mechanisms aligned to psychoneuroimmunology, or PNI, are topics to contemplate across solid organ transplant specialties. In regard to the conclusions, so again, using chemotaxis assays with flow cytometry would aid to give graphical data linking specific endocrines such as adrenocorticotropic hormone or ACTH, and glucocorticoid hormone with the above could be advantageous. Stress may have specific effects with regards to T cell migration post transplantation and migration investigations, instigating 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 pathophysiological migration investigations or studies is one of the most useful ways to one of the most useful ways to look at data to look at data with laboratory investigations and collaborations between transplant immunobiology and neuroendocrinology in context of CKD or chronic kidney disease. It is a challenge to state any specific correlations. That draws this audio podcast to a close. This audio podcast, as part of a Phantom edition. This audio podcast is part of a Vertex Roulet. This audio podcast as part of a Lobostics Machina. This audio podcast as part of a Storm Lobostics production.